Hi, my name's Nicole Lioumis. I am a junior on the Bucknell softball team, and I am a biology major and an econ minor. So this summer I worked in Dublin, Ireland, and I worked in an ovarian cancer research lab where I helped some PhD students look at biomarkers determining early signs of cancer. So I worked 9 to 5 every day, took the bus, got there, and I would meet every Monday morning. We would have kind of a group meeting. Everyone that's involved with the research, we would have meetings, usually about 2 hours long. We would go over all of the people that just got recently diagnosed with cancer. We would go over their scans, their diagnosis, all their symptoms, everything, just to make sure that they were a good fit for our study. We would grab consent from them, and then we would go from there. I mean, being 20 years old, it's kind of hard trying to know everything, especially in a foreign country. I've never been to Europe before, so it was definitely tough, kind of adjusting to society there and how they navigate everyday life. I mean, they take the bus every day. They don't really use cars for the most part, so that was super interesting. But when it comes to work, I think the main thing that I learned was the difference in healthcare systems. I mean, the US is obviously more privatized than it is in Ireland, so it was definitely super interesting to learn how they function, what their rules and regulations are, things like that. I learned how to navigate the lab differently. I mean, they use an entire different metric system, so that was super interesting. But I think the difference in healthcare, just systems in general, was super eye-opening. I mean, it was something that I've never experienced ever. So, I mean, especially working in the American healthcare system, the differences are crazy. For the most part, I worked with the PhD students. They were kind of in the midst of their research, but for the most part, we took the samples from the ovarian cancer patients. We ran them under a microscope. We put them through a centrifuge. I grabbed samples from each one. We put them into different little tubes. We froze them. We took samples even through surgery. I observed a few surgeries. They took some specimens from there and looked under the microscope. I'm not really sure the range of the PhD students' study in general. I mean, I only did so much just because I kind of didn't want to get in the way of them. But for the most part, I ran them through a centrifuge, which is like a huge machine. They just spin it in circles super fast. They spread apart the plasma from the red blood cells, and I just sort of separated them from there. And they're looking at early biomarkers, so like earlier signs, earlier symptoms we can look at through patients' genes to see if we can diagnose them any earlier. The people I met, they're now one of my closest friends, but I didn't know any of them really going in. So I think navigating the whole bus system, the whole working aspect, meeting new people, I think it definitely helped me grow independently, and I think that that will help me down the road with future jobs, future employers, interview processes, all that kind of stuff. So it really opened my eyes to how independent I can be.